Sorry about the interruption. That was about the computer system. Apparently, one of the PDFs that a student sent me that was just pictures of a lab, but it overloaded the whole system, and the system needed to be reset from afar in order for it to work. So I'll have to go after I do this and see if I actually can print the 40 or 50 things that people have sent me since yesterday, that I can then sit down and. It's really tough to mark stuff on a screen. I need stuff on paper to mark things. So we're here and we're moving here. I'm hoping with the microphone that I can wander away and you can still pick me up pretty strongly. Is that was one of the downsides of using the iPad microphone, is that standing over on this side, very often I'd have to, if I was listening to one of the videos, turn the sound up in order to catch it. But hopefully everything will be coming in at the same level now. Okay, here's a huge surprise. Chemical reactions move energy around. And in order to get them started, you got to give them a push. That push is called activation energy. You push, and a lot of times it's just a matter of, the particles are bouncing around, and maybe they'll hit each other the right way every once in a while. But what are the odds? Where if you do something to knock them together more often or in the right way, then you're going to get the reaction to happen a lot faster and a lot more of it. You're going to activate the reaction. Once you get the reaction going, it depends on the type of reaction you've got, what you need to keep doing. If you, that needs to go back in the folder. If you light a match to a piece of paper, that's a chemical reaction. The heat from the match causes the starches in the paper to pop apart. And the energy that used to be holding them together is released, which comes out as heat. You get carbon and carbon dioxide and water. So it pulls the molecules apart. But it releases so much energy that once you get it going, it can keep itself going, right? The paper will continue to burn. The burning of the paper will cause the burning of more paper. That is an exergonic reaction. A reaction that once you give it that nudge and get it going, it can keep itself going. It releases enough energy to keep itself going. Whereas those plants we were talking about before, you flip the light out and stuff is going to grind to a halt. You got to keep pumping energy into a system. And very often the reaction is holding on to energy at the end. So energy goes in. When the reaction is over, a lot of the energy that went in, some of it comes out as heat, but a lot of the energy that went in is holding bits and pieces together. It's stored in there. It isn't being released to keep the reaction going. And that's an endergonic reaction. It does not release enough energy to keep itself going. Now here's another thing that shouldn't be a huge surprise. In living things, very often, if you want to get a reaction going and it's an endergonic reaction, you got to feed it energy. Well, we have chemicals to do that. The breakup of the chemical releases energy to feed this. So one reaction will feed energy into another reaction. One exergonic reaction will feed energy into an endergonic reaction. So the reactions are linked together. They call that coupled. They're coupled reactions. You need to get the energy from one to feed energy into the next one. And this is very often done with ATP, which is, well, So ATP, you can pull a phosphate off of there. I'll show it to you in a second. And that releases a decent amount of energy. That energy gets transferred here. This is your chemical reaction. A is changing into B. So it could be a bunch of things here. They're simplifying this. But when you get done, a lot of the energy that got released is now in here. Now, here's the problem with this drawing. This is a measurement of energy. This thing should be down here. Think about this for a second. I'm hoping that makes sense to you. Because this is going to pick up some energy to be here, but it shouldn't pick up an equal amount of energy as to what this had to start with, right? Energy in this step is going to get lost off. So this one will wind up probably here. Because it'll have energy in it, but not as much energy as was in here in the first place. If that didn't make sense to you, it's not that big a deal. I might ask you in a bonus question about it. And here, adenosine triphosphate is now adenosine diphosphate in a loose phosphate group. What the hell does that mean? ATP is an incredibly useful molecule. It's used in virtually all living things. In fact, I think it's used in all living things. As a let's get this done fuel supply. This string of phosphates, you see this squiggly line? That's a high energy bond. If you break that, you get a decent amount of energy out of it. So you want fuel that you can use for all sorts of things in a cell. What kind of molecule is good for that? Well, you need something that you can break apart and release the energy that's holding it together. Great. We can move that around. And there's a couple of different ways that ATP works. You can actually break the bond and just transfer the energy over. But you can sometimes break the bond and move this onto another molecule. This is a high energy piece. And it destabilizes the hell out of the molecule and gets it to do things that you need it to do. So both of those work. But here's the deal with ATP. It's unstable enough that you can break it apart pretty easily, but is stable enough that you can make it and move it and use it without just having it fall apart on the way. There's not a lot of molecules that fall into that category. Stable enough to be mobile and usable, and unstable enough to be really useful as an energy source. So you got ATP. And we'll talk about how this works in the next chapter. But ATP, it breaks apart into ADP, and pi is just a shorthand for phosphate group. So here's your ATP. It breaks, it releases energy. In order to make more, you take adenosine diphosphate. Now there's just two phosphates. You put a phosphate back in. You feed energy into the system. You can do that through photosynthesis. You can do it through respiration, and there's a few other ways. And so we're always making this and using it, and making this and using it, and making this and using it, because we can do all sorts of stuff that needs energy in our systems. This is our metabolism, right? It would be if I could spell metabolic. Metabolic reactions. Metabolism, tracking the energy through a system, right? We talk about our metabolism. You can talk about a cell's metabolism. You can talk about a mitochondria's metabolism. You can talk about the metabolism of a single reaction. But a tremendous amount of what we do in our systems doesn't move energy from A to B. It moves energy from A to G through B, C, D, E, and F to get there. So there's also what they call metabolic pathways. And it could be two steps, but I'm just going to write down many steps. Between what you start with and what you're ultimately trying to get to, there are a bunch of steps. And it turns out that each step is going to need help in a cell. Is that activation energy for us, for getting things going, for the stuff that we do in a cell, it takes a lot of activation energy, or It takes an approach where it doesn't take as much because you've goosed it a little bit. That's what the rest of the chapter is about. Enzymes. So here's a little trick thing. That nudge that's needed is that in order to get these things to react, you need to nudge them over a threshold. You have to put just enough energy into the system to get them to do what you need them to do. And as you put energy in, some of them will react. And this particular reaction will cause them to lose energy. Is that the product is probably a breakdown product because there's less energy at the end than there is at the beginning. In some cases, you have to nudge them up and give energy to them because there's going to be more energy at the end. Doesn't really matter. Activation energy. What an enzyme does, I don't know if that's going to show up on the screen you're looking at, so I'm going to move this into the middle. Enzymes grab a hold of the reaction molecules. If there's a couple of them, it grabs a couple of them. If it's one and you got to break it up, it grabs one. And once it grabs them, in order for them to react, they need to hit each other just a certain way or stress at a particular bond. Well, if they're putting them together, it grabs them a particular way and doink, puts them together. Doesn't take anywhere near as much free bump energy as it does when you grab them and knock them together. So you knock this threshold down and way more of them will react. If you're breaking something apart, you grab onto it and you stress it and it makes it way easier to break it apart. So enzymes make it so it doesn't take anywhere near as much energy to get stuff to react the way that you want it to react. Okay, I'm going to write some stuff down and then we're going to go back to pictures. Remember way back when we did reactions and what goes in at the front end is either reactants or substrates? And I said that in biological systems, substrates is the more common word. Well, that's because something stuck on something else is a substrate. And most of the stuff that happens in our cells is stuck on enzymes. Something else we talked about way back when is specificity. How specific is the enzyme to the reactants, to the substrates? Will it grab only one particular type of substrate, or will it grab some similar shaped ones? And that can become a pretty big deal. Okay, sucrose is cane sugar. And we have when we take it in, it's two sugars, it's glucose and fructose stuck together. And in order for us to use them, we need them separate. In order for us to really absorb them, we need them separate. If you put them into your digestive system and kind of wait around for them to break apart, they ain't breaking apart anytime soon. You need to get them to separate. So, they fit into the dent on sucrase. Remember ASE, the ending of an enzyme? That's how you write them. And there's a dent shaped like these guys. Now here's why I'm not crazy about this. The act of sticking here will change the shape of this. Remember, an enzyme is a protein. It's a big three-dimensional blob. Well, when you stick stuff into an active site, remember I said it grabs it and it stresses it? When you stick stuff in the active site, it changes the shape of the enzyme, which they're not really showing here. But it does give you a basic idea. where they stress it, this bond that was holding them together is broken, and once it's broken, these things don't stick in there anymore and they come loose. This would be better if it was up here. And then it's ready to grab another one. This goes the other way around, doesn't it? Okay. I get backwards on the screen sometimes. So this comes in and sticks. This changes shape. It breaks the bond. Hydrolysis, you need water to go in to form. You don't need to remember that. It breaks them apart. They come loose. It empties the active site. It's ready to grab another one. And this can go thousands of times a second. These guys can work. If they're in a place where there's a lot of the substrate around, they can work pretty well. Okay, I like this. This one works a little bit better. It's the same basic idea, but it does actually show the enzyme changing shape when this sticks in there, and how once it pops out, it goes back to the original shape. It doesn't really show a stress here, but you know, that wouldn't be that easy to do with a picture. Perhaps if it bent. So you've got all these molecules that make other molecules work. Do you want them doing that all the time? And the answer is no. You want to be able to turn enzymes on and turn enzymes off, because your other alternative is to, every time you don't, excuse me, need an enzyme, just break it down and then rebuild it the next time you need it. But there's a lot of enzymes that it's like, okay, you've done enough. Shut off for a while. We'll turn you on when we need you. That's called inhibiting, you know, because they named it a while ago and they like big words. And inhibition is the process. Inhibitors are the molecules that make this happen. And there's a few different ways. Now here's a weird coincidence. Way back when we did experimental design, we talked about direct observations and indirect observations, and it turns out there were two different indirect observations, using other people's or using devices. Weird thing here, there's one type of direct inhibition and there's two types of indirect inhibition. Other than that, that's the only coincidence. Direct inhibition is okay, I've got the, sorry. I was going to show one picture, but I realized I got a picture that sort of shows this. So here's how this is supposed to work. Here's your substrate, here's your active site. They fit in there, they do whatever they're supposed to do. Here's an inhibitor. And it's in the active site, so the substrate can't get in there. Now you can see a shadow of something I drew in there a long time ago. The inhibitor, this is direct inhibition. It's in the way directly by being where the substrate's trying to be. It doesn't need to fill this whole site up. It can be a little tiny bit here and still keep the substrate out. I like the comparison of you're trying to park your car in a garage. Got to back into the garage. And you start to back up and you look and somebody's left their tricycle in the garage. And you can't get in there. The tricycle's in the way. It's a super hard, not going to move tricycle, and you can't just crunch it. It's directly inhibiting your opportunity to put your car in the garage. That's direct inhibition. Now, in indirect inhibition, this is one way, but I'm going to tell you the other way first. Go back to the garage. You're backing up and you realize that somebody has leaned their bicycle up against, like if this is the garage door, they've left their bicycle so it's kind of hanging in front of the door. It's not in the garage where you're trying to put the car, but you can't get past it to get into the garage. That is one type of indirect inhibition where somewhere near the active site, the inhibitor attaches and it keeps the substrate from getting into the dent the active site. When you're grabbing a big molecule that needs to be, stressed and broken, just a bumpy inhibitor sitting alongside, you can't get that big molecule down into the dent that you need. So that's an indirect. And the competitive part is, are you actually competing for the space or not? Non-competitive, you're attaching somewhere else. You're not actually in the active site. Okay, this again is not the best drawing in the world for what we're talking about. But here's another type of indirect inhibition. There's a spot somewhere on the enzyme, they call it a regulatory site. And when the inhibitor sticks there, it changes the whole shape of this protein blob. Now, what they are trying to show here is it makes the active site smaller. I'd be happier if they had really showed a major change in the shape. They changed the shape of the active site, because that's what's happening here. You change the active site shape, and then the substrate can't fit in there anymore. This is basically inhibition, keeping the substrate from getting into the active site to do what it needs to do. That's a good place. There's a few more things we got to cover, but we're running long here, so I'm going to cut this short. Actually, it's not that short, is it?